Welcome to the unit on evolution. In this unit, we will discuss the process of evolution from a molecular perspective, as we learn to examine the changes in allele frequencies over time. We will be examining how allele frequencies are connected to the genetic variation of a population, and then how to determine if that genetic variation is changing over multiple generations of the population. We will also examine the phenomena that influence allele frequencies of a population, including natural selection. In this first video, we will discuss allele frequencies and genetic variation. In order to understand evolution, we need to first understand some terminology that will be used. When we discuss evolution, we are really examining how populations change over time, or from generation to generation. A population is defined as a group of the same species that is living in the same place at the same time. For example, this could represent a population of frogs living by the side of a pond. Evolution occurs due to changes in the genetic variation of the population's gene pool. A gene pool consists of all the alleles for a single gene in a given population. Remember, from our section on genetics, each individual contains two alleles of a gene, one from their mother and one from their father. Looking at these frogs, some could be homozygous dominant for our gene, some could be heterozygous, and others could be homozygous recessive. The gene pool is the sum of all these alleles in the population, which means if we counted them up, our gene pool consists of 1, 2, 3, 4, 5, 6, 7 dominant alleles, and 1, 2, 3, 4, 5, 6, 7 recessive alleles. Because we see a variety of alleles, both dominant B and recessive B, there is variation in this gene pool. Genetic variation, or a pool of different alleles in a population, is required in order for natural selection and evolution to occur. Interestingly, the difference in genetic variation at the DNA or allele level is not always seen at the phenotypic level. Humans show a high degree of phenotypic variation, as seen in the picture. However, as a species, we actually have a lower level of genetic variation than others. Even though the penguins shown here are uniform in appearance, they are actually 2 to 3 times more genetically variable than we are. This means when you compare the DNA similarity between two people, you will find less differences between them than if you compared the DNA similarity between two penguins. Keep in mind that mutations are the main source of new alleles in the gene pool. Thus, they have an important role in creating genetic variation. For example, if we start here with our population in generation 1, we see no genetic variation because all the alleles are the same. However, if one of those alleles in the germline picks up a mutation, then that mutation will be passed to the next generation. Generation 2 now has 2 different alleles, which increases the variation. Additional mutations can occur in those germ cells each generation, creating more genetic variation. Remember, if the mutation causes a change to the amino acid sequence of a protein, we may also be changing the phenotypic and functional variation in the population. Remember from Chapter 1 on meiosis that we can also increase variation due to crossing over, also called recombination, between homologous chromosomes during meiosis 1. Here's an example of how that crossing over can generate new alleles in the gene pool. In order to measure the amount of genetic variation in a population, you must know the rates of occurrence of the alleles in the population's gene pool. Rates of alleles are calculated by determining the allele frequency, which is the number of that particular allele present in the population, divided by the total number of alleles in the population. If we go back to our population of frogs from earlier, we can calculate the allele frequencies for this population. We had observed 7 dominant B alleles and 7 recessive B alleles. These alleles were out of a total of 14 alleles in the population, and thus we have a frequency of 0.5 or 50% for both the dominant and recessive allele. While the method we just used to count up the alleles and plug in the values was very straightforward, that could be very time-consuming if we had a large population. For example, say we had a population of 100 frogs instead, and we are given the frequency of each genotype in the population instead of the hard counts of numbers of frogs or alleles. To determine the frequency of the individual alleles, we must first find out how many frogs of each genotype are in the population. If 60% of the frogs are homozygous recessive, then that would mean that there are 60 frogs out of the total 100 frogs that are homozygous recessive. Remember, to work with percents, you must convert the percentage to a decimal and then multiply that by your total population. So we obtain 60 frogs by multiplying 0.6 times 100 total frogs. Using that same math, we would find that 10% of the frogs are heterozygous, which means 10 out of our 100 frogs, and 30% are homozygous dominant, so we have 30 homozygous dominant frogs. We can now use this equation to solve for the frequency. The frequency of the dominant allele, which is the percentage of the dominant allele, will be equal to 2 times the number of homozygous dominant individuals, because each one of those individuals has 2 copies of the homozygous allele, plus the number of heterozygous individuals, because each one of those heterozygous individuals has 1 copy of the dominant allele. We will divide that by 2 times the total number in the population, because each one in the population has 2 alleles. Solving this equation would be way shorter than counting the individual alleles for 100 different frogs. Plugging numbers in, we would get 2 times 30 plus 10 divided by 2 times our 100 frogs. This tells us that the percentage of our dominant allele in the population is 0.35 or 35%. You should now pause the video and calculate the frequency of the recessive A allele on your own. This will follow a similar equation. However, instead of having 2 times the number of homozygous dominant individuals, you would have 2 times the number of homozygous recessive individuals. You should have set up your equation to read 2 times 60 plus 10 divided by 200, which would have given you a frequency of 65%. In the previous problems, we were given the genotype frequencies, or the counts of each genotype. But how are the genotype frequencies calculated to begin with? There are 3 ways we can examine the allele or genotype frequencies of a population. The first 2 involve looking at the DNA sequences either indirectly by looking at the proteins using gel electrophoresis, or directly by determining the sequence of nucleotides. The third method is to just look at the phenotype of the population. However, this involves the use of the Hardy-Weinberg equation, which we will discuss in the next video. Gel electrophoresis is a technique that uses electric current to separate molecules, either DNA or proteins, based on their size. In gel electrophoresis, the samples that you want to separate are loaded into holes called wells in the top of an agarose gel. Agarose gel is similar to really dense jello. This gel has pores in it, which small molecules can easily fit through, but larger molecules cannot. When an electrical current is applied, a negative charge is added near the wells with a positive charge towards the bottom of the gel. The negatively charged proteins in DNA will then travel towards the oppositely charged pole, with the smaller ones moving faster through the pores than the larger molecules. After you shut off the current, you can stain the gel for your DNA or protein of interest and be able to locate it by seeing bands on the gel itself. To use gel electrophoresis to determine genotypes of organisms, which can then be used to determine allele frequencies, proteins are extracted from samples obtained from different individuals in the population. Those proteins are then separated using gel electrophoresis. and stained with a molecule that would detect just the protein in the cell that you are interested in. Remember that because alleles are slightly different in nucleotide, they will produce slightly different proteins, and these differences cause them to separate differently on an agarose gel. If an individual is homozygous for a particular gene, then they will only express one type of protein, which will produce a single band on the gel, such as this individual here, who is homozygous for allele 1, or this individual who is homozygous for allele 2. But if an individual is heterozygous for the gene, then they will express two types of the protein, producing two different bands on the gel, such as this individual here. Looking at this example in detail, we can see that we analyzed the genotypes of 8 different individuals for a gene with 2 alleles. The 8 samples produced a gel with 3 different banding patterns. 4 individuals, numbers 1, 2, 5, and 8, had just one band at the allele 1 position. 2 individuals, numbers 4 and 7, had just one band at the allele 2 position, and 2 individuals, numbers 3 and 6, had 2 bands indicating they were heterozygous. We interpret this as 4 individuals homozygous for allele 1, 2 individuals homozygous for allele 2, and 2 heterozygous individuals. From this, we can now calculate the frequency of the dominant allele in the population, just as we did earlier for the frogs. So the percentage of A would equal 2 times our 4 homozygous dominant individuals, plus our number of heterozygotes, divided by 2 times our total population, which was 8, giving us an overall frequency of 62.5%. Similarly, the recessive allele would be 2 times our 2 homozygous recessive individuals, plus our 2 heterozygotes, divided by 2 times 8, giving 37.5%. DNA sequencing allows modern population geneticists to look at any variation in DNA sequence by examining the sequence of nucleotides in a gene. The sequences provide the variation that you use to calculate allele frequencies. For example, scientists sequence a specific gene in 50 fruit flies. This means they are sequencing 100 alleles of the gene. They find that 70 of those have one particular sequence, and 30 of them have another. This would mean that there is an allele frequency of 70% and 30%. The easiest and most typical way for determining allele frequencies is to examine the phenotypes caused by those alleles. However, this becomes problematic for the dominant allele phenotype because you are unable to know how many individuals with a dominant allele are heterozygous or homozygous dominant, and that information changes the frequency of individual alleles. For example, with this population of brown and black labs, the brown labs are all going to be homozygous recessive in order to be brown in color. However, the black labs must have the dominant black allele, but we are unsure whether they would have a recessive brown allele or be homozygous dominant for the black allele. The frequency of the alleles will differ depending on what that second allele is. In order to use observable characteristics, we must use the Hardy-Weinberg equation, which will be discussed in the next video.